This is where we left off. We want to move on to the next slide and talk a little bit about what parasites are. Parasites are the bacteria that can cause disease. We call them pathogens. So those bacteria, and it's not all of them that can cause disease, but those that can, we call them pathogens. And different ways that pathogens can work to cause diseases and some of the diseases that work this way. So bacteria can create exotoxins. And exotoxins can be created inside the bacterium and released into the environment, particularly if this bacterium tends to get into your body. This is how cholera works. They are released, they're made inside, they're released outside of the bacterial cell via vesicles, etc. Or they can be released in terms of endotoxins. And with endotoxins, what these are, the toxins are actually they're actually on the cell membrane itself, and when the cell dies and breaks apart, it will release these endotoxins. They're just basically part of the cell dying. And then the pathogens can actually kill the cells themselves. That particular one isn't in your textbook, but certainly exotoxins are released from the bacteria outside of the bacteria. Endotoxins is when the bacteria is dying, portions of its plasma or cell membrane are considered toxins by the body, and that is how plague works. Killing cells off itself is how influenza works. And then last few topics is the ability or the positive nature of prokaryotes. We talked a little bit about their negative nature, but there's great understanding with regard to positive nature. Certain DNA technology has been created. This idea of CRISPR has been created because of prokaryotes. Prokaryotes have a repeating unit of their DNA, and prior to that unit you have a specific type of protein. They call this the CRISPR-Cas system. The Cas part is a protein that repeats, and so they actually can find where this CRISPR sequence is and use this idea of repeating sequence to actually insert a DNA sequence into a particular cell. So use this for an insertion technique. They're using this very actively in terms of research to try to fight HIV. And so it fights certain viral infections that can cause death in humans. So DNA technology, bioremediation. So prokaryotes are being used to break up oxygen or break up oil spills. So those petrochemicals, they actually feed on the petrochemicals to break up these oil spills. And then also mining and synthesis of certain things that we use, like vitamins, antibiotics, hormones, and other products. The image that you have, what do these products have in common? All of these foods use prokaryotes to be created. Cheese, tofu, which you probably knew cheese. Tofu uses prokaryotes. Soy sauce uses prokaryotes. Beer and wine, prokaryotes, and also chocolate. You need bacteria to make them. And sauerkraut, any fermented food is going to use a bacteria to create it. So many positive uses for prokaryotes also. And then here's your list of to knows: gram-positive, gram-negative, certain terms to get in your head, F factor, R plasmids, and then distinguish between other terms in the chapter. That's all we have for this one, and we will move on to chapter 28.